the National Institute of Neurological Disorders and Stroke, NINDS, a part of the National Institutes of Health. Phosphorylation is a process where a phosphate ion is added to a specific amino acid, or building block, of a protein, in this case the protein α-synuclein. This addition can change the shape of that protein, causing it to change its level of activity. Most studies of phosphorylated α-synuclein have studied its role in certain neurological disorders such as Parkinson's disease and Lewy body dementia, where it builds up in protein clumps called Lewy bodies. These clumps are thought to be toxic to neurons, and one of the prevailing hypotheses is that the phosphorylation of the protein α-synuclein triggers these diseases. In most studies to date, the mere presence of α-synuclein phosphorylation is assumed to be a marker for pathology for certain disorders, like Parkinson's and Lewy body dementias. Recently, there has been considerable interest in developing drugs that prevent α-synuclein phosphorylation as a way of treating these disorders. These findings challenge the current hypotheses about how these disorders may originate in the brain and may give insight into how we might better treat them. Using a molecular modeling strategy to look at the structure of α-synuclein, it is discovered that when α-synuclein is phosphorylated, its structure changes in a way that promotes interactions with other proteins in healthy brains. Furthermore, they observed an association between increasing neural activity electrically or chemically and an increase in the amount of phosphorylated α-synuclein in both cultured cells and in mouse brain tissue. This finding suggests there might be a relationship between synaptic activity and α-synuclein phosphorylation.